Hello, my dear students. Welcome to Shri Jayendra Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology section. My dear students, we have started evolution chapter in the last lecture. So please refer and go through the last video so that you can have a better connect for the upcoming videos. In the last lecture, we came to know that how different forces they enable the primitive conditions on this planet for the evolution and for the origin of life. We have learned about the most famous Harold Urey and Stanley Miller experiment, which proves that life originated from the simple molecules. These simple molecules, they are converted into simple organic molecules. Then these simple organic molecules are converted to complex organic molecules. And in this way, the organic compounds which helps to support the life like carbohydrates, proteins and fats, they are synthesized. Now we have discussed about that how the protoplasmic matter is to be formed. In the protoplasm, it requires all these essential substances, including water. So now we are going to discuss about that how biological evolution takes place. Till we have studied the evolution of inorganic materials to organic materials. Now we are going to discuss about the biological evolution, that is the organic evolution. That is the organic evolution. Right? Now, the most important thing is the moment we are going to study about the biological evolution or the organic evolution, we have to connect every frame what we have studied in the previous lecture. That how when we studied about the chemogeny, the atomic stage, the molecular stage, everything. So please have that connect. Now, origin of protobionts. Proto means primitive, bionts means living or like a living cell. So origin of protobionts and nucleoproteins, also known as conservates, they were the macromolecules which were synthesized abiotically in primitive ocean. I mean, they were the first form of primitive life or primitive cell which are synthesized abiotically in the primitive ocean. Later, they came together and formed large colloidal drop like structures known as protobionts. So, origin of protobionts and nucleoproteins, that is, co-surfates, how they are originated. Macromolecules which are synthesized abiotically, the macromolecules like the carbohydrates, proteins, fats, nucleic acids, everything which is formed abiotically in the primitive ocean, in the prebiotic soup, in the prebiotic ocean, they came together and formed large colloidal drop-like structures. They form a large colloidal drop-like structures named as protobionts. So in this way, protobionts is an assembly of the macromolecules which were synthesized during the chemical evolution. These protobionts, they were known co-surveys by Oparin. So protobionts, they basically exist in two morphological forms, either co-surveys, which was given by the Oparin concept, and the next is microsphere, which was given by Sidney Fox. Okay. So according to Sidney Fox, there are around Formation of non-enzymatic proteins, non-enzymatic proteins, which are short nucleotide sequence. Okay, with around 18 amino acids, they form the very first protein structure. Okay, very first protein structure. Suppose that protein structure is P1. Okay, so according to Sidney Fox, the non-enzymatic protein, they basically make a 18 amino acid collaborated structure, which is the first protein which was non-enzymatic. Okay, so according to Uparin, these protobionts, they are actually coservates, and according to Fox, they are microspheres. And according to Demer, they known to be vesicles or vesicles, right? So, these three different forms in which the coservates and the microsphere, they are the most prominent. Okay. So, these protobionts or protocells, they don't have any biological membrane. Okay. When they are having a non-biological membrane or an enclosure, that is why they are known as what? Protocells. Mean they have a boundary, and if that boundary is present, that boundary should be non-biological. Okay. So these protocells and protobionts, they basically existed for a very short time, very short period, okay? And they can produce further, they can produce membrane bound vesicles known as protocells. So it was protobionts, which was just a mixture or a collection of, I can say it is a collaboration of macromolecules having a non-biological membrane. When these protobionts, and especially speaking, these protobionts or the primitive forms, they don't have capacity to duplicate. They don't have capacity to replicate. They don't have capacity to divide. So when these protobionts, they experimentally lead to the production of membrane-bound vesicles known as protocells. So basically, Halden and Fox and Demer, they actually created some kind of artificial, you know, requirements. Okay, they proved that these coservates, microspheres, vesicles, they actually exist or they can exist in the artificial environment, which will give us a sense that how life is being originated. Okay. So first we will study about co-surveys. So co-surveys, the concept was given by Oparin. It was basically a hypothetical, concept which was proposed by Oparin in the year 1920. According to this, the early form of life means the protocell, they may exist in a form of co-survey. Oparin gave the term co-survey. Okay. So basically, if you got to know that Oparin, he proposed the term co-survey, and these co-surveys, they are non-living structures. Co-surveys, they are non-living structures. Non-living structures. Okay. That lead to the, or that basically help in the formation of first living cells from which the more complex cells arises. Okay, mean evolved. So that was the concept which was given or proposed by Oparin, that the life first existed in a form of coacervate, which is basically a non-living structure. These non-living structures, they can give rise to the living forms. Understood? So basically, these core surveys, they are a collection of organic molecules. Okay, means they are protobions. They are the collaboration of macromolecules which are basically surrounded by a non-biological membrane. That is, maybe it is a membrane made up of water. Okay, a water film. Okay, so this is a core survey. Each protobiont was a cluster of macromolecule. They contain proteins, nucleic acids, lipids, polysaccharides, all the macromolecules, all the essential, requisites for formation of these protocells or protobionts were present. They grew by absorbing molecules. They have an inherent capacity to absorb molecules from their environment. They can absorb molecules from the environment, right? They could divide by budding like bacteria. Now they come up with, means the protocells or the coacervates, they come up with the process of division, but the division is very simple type budding, like bacteria. Many chemical reactions, including decomposition of glucose, took place inside the protobionts. So they basically show some signs of metabolism. But as no proteins of those time, they were enzymatic, so signs of metabolism remains in a question mark. Okay, it is likely to see that okay, there are some simple reactions which can be traced, okay, which are confirmed like decomposition of glucose, but we cannot say that they are metabolically active, especially the protobionts or the core surface. They are non-living structures, right? The sun provide energy for chemical reaction. Sun basically give them energy for chemical reactions, and this energy only helps to break the bonds. So that might be the reason that these own cells, they might be not metabolically active, but because of the solar radiations, because of the high gamma exposure and the cosmic exposure, the bonds can be cleaved and new bonds can be formed. According to apparent co-surveys, the first sole living molecules which give rise to cell. Now, protobionts, these which are the collaboration of macromolecules, which are exactly the non-living structures, but according to Oparin, they may convert them or they may have the potential of living co-surveys and they can form the living cell. Okay. So co-surveys, according to Oparin, they were first considered as the living structure, okay, or living molecules which give rise to cell. Exactly speaking, co-survey itself, it is a non-living structure, but it is having some specific molecules which can give rise to the living form. Okay. So these were the protobionts or the co-surveys. You have to remember co-surveys, they were the non-living structures which are basically, having macromolecules surrounded by a film of water. Okay. In a non-biological film. Understood? Now, I am going to discuss about different types of protobionts obtained by scientists. Oparin 1924 took a solution of carbohydrate and large protein. The solution was shaken. It caused separation of co-circuits. Okay. So Oparin, when he took a solution of carbohydrate and large protein, and the solution was shaken, it caused separation of co-circuits. Co-circuits, higher concentration of protein, carbohydrate were present with small amount of water. So basically, co-circuit is nothing. It is just a collection of macromolecules. Okay, especially the proteins, carbohydrates, and some amount of water. Oparin's co-surveys could grow and exhibit simple form of metabolism. Now, according to Oparin's co-surveys, later they come up with simple decomposition reactions of glucose or some signs of metabolism. Okay. However, a lipid membrane and reproduction was absent. I mean, there is no mitosis, no replication, no duplication. Lipid membrane was also absent. Okay. So apparent co-surveys, they could grow, they could enlarge in their size, but they cannot divide. Okay, or they can divide by the simple process of budding. Okay, you have to remember Cocervids, they don't have inherent capacity to divide with no mitosis, no reproduction. If they can divide because of getting into enormous size, and that is why they lead to budding, right? However, a lipid membrane and reproduction was absent. Fox in 1957 obtained microspheres with a primitive membrane. He heated a dry mixture of amino acids at 130 to 180 degrees Celsius. So Sidney Fox, he, according to him, that microspheres with a primitive membrane, okay, I mean microspheres with a primitive membrane means Sidney Fox, according to him, that a membrane bound structure could only be given a post of being a cell. So according to Fox, microspheres, they were the primitive membrane bound structures, and they, when he heated amino acids, a dry mixture of amino acids at 130 to 180 degrees Celsius, and it forms proteoids or proteinoids, means a polymer of amino acid, but it is not a protein. It is a primitive form of protein. That is why it is known as what? Proteoid or proteinoids. As these proteoid or proteinoids, they only have 18 to 20 amino acid sequence. That is why it is known as what? Proteoid. Understood? I'll write it here. Sequence of 18 to 20 amino acids. That is why it is known as what? Proteoid or proteinoids. When the proteoids poured in cool water, okay, mean when these proteoids, they poured in cool water along with lipids, microsphere gets separated, size 1 to 2 micrometers. Okay. So when these proteoids, means short sequence of amino acids, polymer of amino acids, when they are poured in cold water, okay, when they are poured in cool water, then they come up with, some kind of precipitation, okay, and lipids, along with lipids they form microspheres, okay. Dimer, microsphere and co-surface could fuse to form protobions, have various type of chemical like proteins, nucleic acids, carbohydrates enclosed inside a lipid membrane, and according to Dimer it is was known as fascicles. Okay, so it is all about the different, projections of different scientists. Somebody says it's a co-surveyed, somebody says it's a proteoid, somebody says it's a vesicle. Okay, the main thing you have to remember that the formation of microspheres, they exactly bring a sense of revolution, as the collection of macromolecules is now basically having a membrane, and this is what required by those time for the formation of protoplasm. Exactly, yeah. And that is why the proteoids formation or the proteinoids formation is supposed to be, microsphere formation. It's supposed to be, come up with different types of metabolic reactions as well. Okay, like for example, these microspheres, they started to show some simple metabolic reactions, and they started to show some simple means of motility as well. Okay, some simple metabolic reactions and some simple motility as well. Okay, in case of microspheres, right? These microspheres, it is supposed that these microspheres, they basically, they don't have any scope of diversification. Okay, so when we talk about that, okay, protobionts means, suppose they are the collection of membrane bound these structures, okay, and these membrane bound structures, they don't show any scope of diversification. No scope of diversification. Okay, means as they are not showing any scope of diversification. Okay, so these microspheres, they are actually, you know higher, or they are evolved than protobionts. Okay, and that is why they are not showing any scope of diversification. Okay, now this is clear. Now moving to the next slide, origin of protocells or eubionts. So it was the formation of now origin of protocells or eubionts. The first living form named protocell originated in the primitive oceans. Yeah, that's very clear. We have just gone through the same statement. Okay. The protocell were the cluster of nucleoproteins. That's very true. The protocell were the structure, so having clusters or the collection of nucleoproteins. Now, what exactly nucleoproteins? Okay, nucleoproteins, they are basically assembly of nucleic acids which are attached to proteins. So these nucleoproteins, they come up with the very first sign of life, means the formation of genetic material. So the protocell were the clusters of nucleoproteins which formed composition of nucleic acids and enzymatic proteins. So these protocell, they are actually clusters of nucleoproteins which form the composition of nucleic acids, okay, and enzymatic proteins. Very important, enzymatic proteins. But now these proteins, they can undergo different types of catalysis or different types of metabolic reactions. Okay, so protocells, the very first, they started to show the signs of metabolically active. They are having nucleoproteins. Nucleoproteins, they are basically the composition of nucleic acids and enzymatic proteins. Okay, nucleoproteins had the property of self-duplication. Okay, nucleoproteins, they had self-duplication property. So please remember this term. The nucleoproteins, they had a property of self-duplication. Nucleoproteins, they were the first sign of life. Nucleoproteins, mean, uh, they were thought to be the first sign of life. Protocells represented the beginning of life. Protocells, it represents the beginning of life, right? From protocells or eubionts, few core of nucleoproteins get separated, free in oceans, and become inactive. But when they enter in another eubionts, they become active. So virus-like structures were formed. Okay. From protocells or eubiont, there were some nucleoproteins which get detached and separate, and they set free in the oceans and become inactive. But when they enter in a particular or another eubiont, they again become active. So this basically can be a sign of viral transformation or virus. Origin of virus-like structures is an example of retrogressive evolution. Retrogressive evolution means from complex to simple. So viruses, they are basically formed from protocells, and exactly speaking, viruses, they only contain the nucleoprotein part. And that is why the virus till date, they can only express through their nucleoprotein, means they can only express themselves through their RNA or DNA. They don't have any cellular machinery. And that is why if a protocell giving rise to a viral like structure or virus like structure, it shows that our evolution is going on the backtrack. Okay, it is going from complex to simple. So that is why it is mentioned here, retrogressive evolution. You have to just remember that formation of viruses from proto cells or eubiont is considered to be a retrogressive evolution. Okay, see, when we talk about the microspheres, okay, or the core surveys, especially the microspheres, they are basically made up of proteinoids. Okay, proteinoids, they are just collection of the proteins, amino acids, polymers of amino acids. And proteinoids or proteoids, they are exactly aggregation of these essentially required amino acids. And these essentially required amino acids, they basically give us a history of 3 billion years back. Okay, now they are going to form different other molecules like RNA, proteins, polysaccharides, and other complex structures. So that is why the formation of microsphere is considered to be a evolutionary break 3 billion years back. Okay, that if microsphere they were not formed, so protocells will they were never be existing, because microspheres only they come up with, you know, conditions, they come up with that strange adaptability in which they can survive in that extreme environment of earth of those times. So that is why microspheres they come up with, you know, for the formation of protocells, microspheres were required very much, I should say. Okay. So these were the important points. What you have to remember is that nucleoproteins were the first sign of life. Okay, nucleoproteins they had property of self-duplication, and protocells they are basically formed by microspheres. Microspheres they are known as evolutionary break 3 billion years back. Okay, so the metabolic reactions were first seen in microspheres, then it is seen in the protocells. Okay, enzymes now they are the proteins they are now maybe enzymatic, mean the enzymatic action, the catalysis action comes to be in existence in case of protocells, right? Now coming to next, this is the diagram of co-servate or a protocell. Okay, I have shown to you. You can see that it is having a boundary, and this boundary is a non-cellular boundary, which may be a, you know, a film of water or anything, and inside this they have just a collection of molecules. These molecules they come up with different types of formation of polymers, like you can see here they are basically giving rise to nucleic acids. Okay, and the process of division is very simple. Like for example, the microspheres they divide just like a bud formation. Okay, they make a bud. The most importantly that microspheres they come up with membrane, and that is why now they can protect them. Okay, so this is what about the co-servates or the protocell is. This is proto, I should say a protocell. Mean uh, not a protocell. I should not write it like protocell. A co-servates, apparent co-servates, protobiont. Okay. Now these protobionts only they are converted into protocells. Okay. Now we are going to study about origin of prokaryotes. Prokaryotes, pro means primitive, karyon means nucleus. Khurana in 1970, Indian-based scientist artificially synthesized 77 nucleotide RNA molecule outside a living cell. He synthesized a RNA molecule which was having 77 nucleotides. Okay, which suggests that probably RNA was the first or the most primordial genetic material rather than DNA. Okay, so according to Khurana in 1970, he synthesized 77 nucleotide RNA molecule outside the living cell, which basically suggests that okay, RNA was the most primitive material, genetic material, and it was not DNA. Zung, Thomas, and Altman described that some RNA molecules have enzymatic activity as well. Some RNA molecules have enzymatic activity as well. Hence, probably the RNA enzymes called ribozymes. So, all enzymes are proteinaceous in nature except this RNA or ribozyme, it is not proteinaceous in nature. It is not proteinaceous in nature. Okay? So, I'll write here ribozyme is not proteinaceous in nature. not proteinaceous. Okay, exception. Were able to replicate the primordial DNA. The discovery of RNA molecule working as enzyme has also changed our thinking about origin of life. So, Kurana, Zeng, Hammers, Altman, these different scientists, they come up with the fact that okay, RNA molecules, they could perform enzymatic activity. Okay, and they are known as what, ribozymes, and they were helpful to replicate primordial RNA. The discovery of RNA molecule working as enzyme gave us an opportunity to look forward with the hereditary material and the process of duplication of protocells. Of, it is now believed that about 4 billion years ago, Earth was an RNA world, in which RNA molecule carried out all the processes of life without the help of either protein or DNA. By this discovery, evolution is termed as RNA world. Okay. It is estimated that life originated around 3 billion years ago as protocell eubions in pre-Cambrian era, which was anaerobic heterotrophic. So pre-Cambrian era was anaerobic heterotrophic, which gave rise to the first anaerobic protocell, that was eubion. Origin of prokaryotes. So please remember the distinction between prokaryote and the eubions. Okay. As a result of mutation, mutation means sudden change in DNA. The protocells became more complex and efficient and used the materials available in the surrounding medium and condensed themselves into prokaryotic cells. So these protocells now, because of some changes in their DNA, because as they are having nucleoproteins in them, so now they, because of changes, mutation in their DNA or genetic material, it may be DNA or it may be RNA or it may be nucleoprotein. These protocells, they come up with drastic changes, that is, now they are basically heading towards being more complex and efficient and use the materials around them for their survival. Okay, they condense their size and now they are basically converted into prokaryotic cells. Thus, the first living being were prokaryotic. Okay, when prokaryotes, we have 100% confirmation that they are living, and that is why till date, out of, I mean, outbreaking all the hypotheses, all the assumptions, all the presumptions, prokaryotes, they are considered to be the living, first living being, okay, like bacteria, which were single celled and consisted of naked DNA. Consisted of naked DNA. Nutritionally, they were chemoheterotrophs. Chemoheterotrophs means they can break the complex chemical compounds to obtain energy. Okay, they were chemoheterotrophs means they can break the complex organic compounds to process the energy. Saprotrophs means they feed on the organic matter, because prebiotic soup, the hot dilute soup, was enriched with many macromolecules, organic compounds, and that is why they are feeding on these. Okay, and they were either chemo heterotrophs or saprotrophs. Respiration was anaerobic. Respiration was anaerobic. So nutritionally, it was considered as the first evolved prokaryote. They were either chemoheterotrophs or saprotrophs, because respiration was anaerobic. There were no free oxygen those time. Okay, understood? They simply divide by binary fission. These prokaryotes, they simply divide by binary fission, right? Now, some of the oldest archaeologists under the study of paleontology, means the study of fossils, some of the archaeologists, they excavated some parts of stromatolites, stromatolites, and these stromatolites are formed today from sediments, mean from the deposition of the rock surfaces, the because of the weathering of rocks, because of the soil particles, and photosynthetic prokaryotes. Because photosynthetic prokaryotes, they are basically having filaments, and these filaments, they actually help to bind the soil particles. Okay, so these stromatolites, they basically give us a sign. Okay, the soil particles, the density, the dilute soup is now becoming tensile. Why? Because of the soil, because of the rock particles, because of the minerals. So all these particles, they basically, because of this filamentous cyanobacteria, it attaches to themselves. Okay, so stromatolites, they were the first sign or the most primitive considered to be prokaryotes. Okay, they basically contain filamentous blue-green algae. Understood? Now, moving to the next slide, origin of autotrophism. Okay, okay. Till here we have seen that how prokaryotes they came into existence. Now we are going to talk about the evolution of mode of nutrition. That how mode of nutrition is being evolved. Okay. Under this, the very first is the formation of heterotrophs and then formation of autotrophs. So we are going to study about origin of autotrophism. Before going further, I'll like to tell you about the origin of heterotrophs. The earliest prokaryotes were chemoheterotrophs, as you have seen, either saprotrophs. They basically obtain energy from the fermentation of organic molecules from the sea broth in oxygen-free atmosphere. Okay, so they basically obtain energy by the fermentation of organic molecules deep down the sea in the reducing atmosphere. They require ready-made organic material as food, and thus they are heterotrophs. So earlier plants were chemoheterotrophs. Okay, earlier plants, sorry, earlier prokaryotes. They were heterotrophs. Hetero, H-E-T-E-R, heterotrophs. Understood? Now comes the concept of autotrophism. It includes the origin of chemosynthesis and photosynthesis. Okay. So origin of autotrophism simply means that they may be chemoautotrophs or they may be photoautotrophs. The origin of chemosynthesis. Due to continued withdrawal of organic molecules by chemoheterotrophs. Chemoheterotrophs means heterotroph bacteria, saprotroph bacteria. They are just basically using or withdrawing or exploiting the organic molecules. Organic material decreased in oceans. Organic material it decreased in oceans. Before the organic material disappeared in the sea, new modes of nutrition developed. One of them was chemosynthesis. Okay. So after chemoheterotroph comes chemosynthesis. The organisms which perform chemosynthesis are called chemoautotrophs. The organisms which perform chemosynthesis are called chemoautotrophs. They were anaerobes and synthesized organic molecules from inorganic material. Okay, they synthesize organic molecules from inorganic material. So now they are synthesizing the required organic molecules by inorganic material. It means they are synthesizers or they are autotrophs. The energy was obtained by oxidizing inorganic materials present in the sea. Such mode of nutrition is found in bacteria like iron sulfur bacteria, nitrifying bacteria. So the energy was obtained by oxidizing inorganic materials present in the sea. These inorganic materials, when they are converted into the organic, okay, the energy is liberated, and that energy is basically used by these bacteria, right? Example: sulfur bacteria or nitrifying bacteria. Origin of photosynthesis. After some time, bacteriochlorophyll developed in some autotrophic bacteria like organisms. They could absorb solar energy and convert it into chemical form. These organisms called photoautotrophs. So now after that, the formation of bacteriochlorophyll takes place, which basically gives a sign of formation of photoautotrophs. They utilize solar energy in synthesizing organic compounds. The process is called photosynthesis, right? They were anaerobic and utilize hydrogen from sources other than water, like H2S. Very important. So basically, during the process of photosynthesis, water is not utilized, and if water is not utilized, then there will be no free oxygen. Okay. So H2S gas basically combines with the other materials like carbon, like hydrogen, ठीक है ना, to form different types of organic molecules. Okay. So as there was no water, there will be no oxygen. Okay. So instead of water, a reducing gas was there, H2S. H2 was an oxidizing gas. Therefore, no oxygen was involved and atmosphere remains reduced. The stage of photosynthetic autotrophism is represented by planktonic sulfur bacteria of today. Okay. Stage of photosynthetic autotrophism is represented by a planktonic sulfur bacteria of today. If today, especially when we talk about the planktons, okay, so these planktons, they basically have, a assembly of sulfur bacteria, okay, and these planktonic sulfur bacteria, free floating, small plants or plant herbs, they have assembly of sulfur bacteria, okay, maybe in a symbiotic relationship or maybe colonized, and these planktonic sulfur bacteria, they are considered to be the photosynthetic autotrophism, signs of photosynthetic autotrophism. Okay, so I hope, is it clear? So the main things you have to remember that first the earlier prokaryotes were heterotrophs, then they become chemoautotrophs. Okay, and these chemoautotrophs, they help to break the chemical compounds and to make the complex compounds. Okay, so they basically form inorganic. They basically form the organic compounds from the inorganic materials. Okay, they were anaerobic. Example: sulfur bacteria and nitrogen bacteria. Then the origin of photoautotrophism or photosynthesis came into existence because of the formation of bacteriochlorophyll. Was this bacteriochlorophyll like bacteriovertin? Developed in some autotrophic bacteria like organism, they could absorb solar energy and convert it into chemical form. These organisms called photoautotrophs. Okay, as the you can see that there was no water which was utilized, only there was H2S. That is why there was no free oxygen in the atmosphere. The atmosphere was reducing. Okay. Now coming to next slide. Evolution of mode of nutrition, already I have told you. The bacteriochlorophyll by molecular changes formed a true chlorophyll. So this bacteriochlorophyll comes up with different type of changes and form true chlorophyll. And such organisms which were bearing true chlorophyll are similar to today's cyanobacteria, that is blue, green, and green. They utilize water as a hydrogen donor and evolved oxygen. Previously, the hydrogen donor was H2S. Okay, and here the hydrogen donor is the water, and that's why the free oxygen starts to liberate it. Enormous amount of free oxygen liberated, which forms our atmosphere to become oxidized. Oxygen revolution. Liberation of free O2 by blue-green algae like prokaryotes due to the photosynthesis was a revolutionary change in the history of Earth. Liberation of free O2 by cyanobacteria, blue-green algae, okay, like prokaryotes due to the photosynthesis was a revolutionary change in the history of Earth. And now the atmosphere of Earth becomes oxidizing. It is called oxygen revolution. It includes important changes like atmosphere of Earth changed from reducing to oxidizing, hence possibilities for further chemical evolution. Previous the organisms, especially the prokaryotes, whether we talk about the chemoheterotrophs or the chemoautotrophs or the photoautotrophs, they were metabolically less active because oxygen was not available. The moment oxygen starts to available to these organisms, they become highly active, metabolically active. Okay. The possibilities of further chemical evolution and abiogenesis got over. Okay. Because chemical evolution always take place in reducing environment. Always remember that chemical evolution always take place in reducing environment. Okay. The atmosphere of Earth changed from reducing to oxidizing, hence possibilities of further chemical evolution and abiogenesis got over. Okay. Now, because chemical evolution always can only take place in reducing, and now we have oxidizing environment. So now the possibilities of chemical evolution were very less. Free O2 oxidizes methane, ammonia to form gases like CO2, nitrogen, and water. Okay. Accumulation of free O2 formed a layer of ozone above the atmosphere of Earth, which started absorbing most of the UV radiations of sunlight and blocking the cosmic rays as well. Okay. So the moment oxygen came into existence in our atmosphere, these were the following changes. First, the atmosphere of Earth changed from reducing to oxidizing. So possibilities of further getting into any other stuff, further getting into any other thing. Okay, the possibilities of that chemical change. Okay, chemical evolution. Okay, drops down. Abiogenesis drops down, and now the life is going to form. Okay, right here till here only chemicals are evolving. Okay, organisms they are not evolving or their evolution was very slow. Now the chemicals they basically are going to form certain substances which are very essential for the formation of the complex forms of life. So free O2 oxidized CH4, ammonia to form nitrogen gas, CO2 gas, water. An accumulation of free O2 in the form of ozone above the atmosphere of the Earth started to absorb UV radiations of sunlight and cosmic rays burst. Now coming to next, origin of prokaryotes. Origin of eukaryotes is basically related with the origin of prokaryotes. So the line is like this: protobionts, conservates, microspheres, protocells or eubionts, then prokaryotes, and then the eukaryotes. Origin of eukaryotic cell. About 2.7 billion years conditions become suitable for aerobic respiration with release of O2. So around 2.7 billion years ago, the condition becomes suitable for aerobic respiration with the release of O2. Aerobic respiration yields about 20 times more energy than anaerobic respiration. Hence, the prokaryotes adapted themselves for aerobic mode of respiration. Means from anaerobes to aerobes. Nucleus, mitochondria, and other cell organelles developed in a cell. Nucleus, mitochondria, and other cell organelles developed in the cell. And free living eukaryotic cell like organisms originated about 2 billion years ago in primitive ocean. Please remember, nucleus, mitochondria, and other cell organelles developed in the cell, and free living eukaryotic cell organism originated around 2 billion years ago. Okay. Organic evolution, though life originated by chemical evolution on primitive Earth, was later replaced by organic evolution. Okay. First is chemical evolution, now it is organic evolution. Organic evolution states descent with modification, Charles Darwin theory. The present day complex organisms have evolved from the earlier simpler organisms, okay, but gradual changes which have occurred over millions of years. Okay. Though living organisms show diversity in size, structure, function, behavior. They also show basically some metabolic processes indicating some common ancestry. So that what Darwin's believe in. That okay, all these common ancestors they are going to form these existing today's life forms, and that is why it is considered descent with modification. Okay. So the details of this I will be telling in the next class. Okay. This is the evolution of eukaryotes. I will be explaining in the next class with the further different mesmerizing concepts in evolution as well. Okay. So we will be doing a neat time as well in the next class. Till then, take care. Bye bye. Stay safe, everyone.